So as far as I understand, so far what you guys have covered to this point is cellular and molecular biology from a more general perspective, but today we're going to pivot towards trying to study cellular and molecular immunology. And the way the immune system is broken down, there's two sides. There's innate immunity and adaptive immunity. So today we're going to focus on innate immunity, and more specifically we're going to look at it through the context of Crohn's disease. So Crohn's disease is a type of inflammatory bowel disease. There's actually two types of inflammatory bowel disease. There's Crohn's disease and ulcerative colitis. I can put those up on the board. This is not necessary, but as you can imagine, ulcerative colitis is inflammation of the colon, so it's restricted to that part of your GI tract. So it's only in the colon. Where Crohn's disease affects the entire GI tract. And the symptoms actually reflect that. So the systemic symptoms include growth retardation, fever, and weight loss. And so this is generally attributed to malnourishment. And so a lot of the nutrient uptake is occurring in the small intestine, and so that that's not going to be affecting the colon as it is with ulcerative colitis. So generally, Crohn's disease is considered far worse than ulcerative colitis, but ulcerative colitis is still very problematic. And the symptoms that are specific to the GI tract include abscesses, and so there's a colonoscopic image over here to the right, and these yellow lesions here are these abscesses. They're pus-filled lesions. What goes on during Crohn's disease is that there's a lot of neutrophils that are recruited to these sites of inflammation in Crohn's disease. And what happens is, as I'm sure you guys know from your pre-lecture that all of you took very careful notes of, I imagine, neutrophils will be recruited very quickly and they'll act very quickly, but then they also die very quickly. And so they'll usually only live for on the order of a couple days, and they're a phagocytic cell, meaning they engulf anything they can that they can recognize, and they'll just die. And so what happens during Crohn's disease is that they continually are recruited and dying at the site of these lesions. And so it creates a lot of pain and discomfort. There's also a lot of diarrhea. So these lesions also occur in the large intestine, and so that is going to affect water uptake. And so it's going to create a lot of diarrhea issues, and it also creates ulcers. And when these ulcers become so problematic, it can create fistula, which are almost these tubes that connect the GI tract to the outer cavity. And so it's basically like a hole in your GI tract, and that can create a lot of issues that can lead to sepsis. And so another distinction between Crohn's and ulcerative colitis is that ulcerative colitis will only affect the top layer of the GI tract, whereas Crohn's disease can affect the entire thickness of your GI tract. So again, it's a far worse disease relative to ulcerative colitis. Again, some other systemic symptoms include anemia as a result of some blood loss, and then neurological issues. Crohn's disease is also connected to your microbiome, and there's a lot of research going on nowadays that look at the connection between your gut microbiome and neurological issues, and that's manifested in Crohn's disease. This disease, inflammatory bowel disease, IBD, affects about 1.5 million people in America, and half of that, it kind of splits down the middle between Crohn's disease and ulcerative colitis, and that breaks down to about 750,000 or so. And as it is now, there's about 1 to 10 cases per 100,000 people in the United States. So the incidence rate is increasing. And what's also interesting is that there's a bimodal distribution. What that means is that if you look at disease occurrence and age, what ends up happening is that kids about 10 to 20 have a high incidence rate, it breaks back down, but then in older age, around 50, comes back up. So it's bimodal. We don't really know why that's the case, but it's something we've just observed clinically. So the causes for this disease. As with a lot of immune diseases, they're complex, and they also involve some sort of genetic mutation or polymorphism. that enhances a person's likelihood of developing Crohn's disease. What's unique about Crohn's disease is it kind of involves both the innate and adaptive immune systems in the etiology of this disease. So there's a mutation in your innate immune system, and what ends up happening is that your adaptive immune system tries to overcompensate, and as a result of that, your adaptive immune response is chronically responding to your native gut microbiome, and it's killing those off, and it's allowing for pathological microbes to persist. And so it's unique in that it involves both arms of the immune system. There's been a lot of work that has focused on a specific mutation that's relevant to this lecture, and it involves a variant in NOD2, and we'll be talking about what NOD2 is later in this lecture. And so it connects innate immunity to Crohn's disease specifically. So you guys in the pre-lecture looked at some of the important qualities of the immune system, and what we're going to do now is we're going to kind of go over those in light of the innate immune system specifically. Diversity. So in the innate immune system, there's a lot of different features that your innate immune system can respond to. Now, it's going to be different than adaptive immunity, and we'll touch on that later. But there's so many different kinds of pathogens that your body can respond to as a result of your immune system, your innate immune system, that has been evolutionarily conserved over generations. And while you can respond to a wide array of antigens, there's also the ability for a single receptor to recognize a specific pathogen or a specific antigen on that pathogen. And so the specificity in light of the diversity is really what makes the immune system quite powerful. And memory. Memory is really going to be important for adaptive immunity, but it's also relevant for innate immunity because, as I mentioned earlier, the pathogens that you respond to have been evolutionarily conserved over generations, where we've kind of co-evolved with the microbes that our body is able to respond to, and we've recognized them over many, many, many years. And so we're going to break down the segments of the immune system into two specific categories: humoral immunity and cell-mediated immunity. Humoral immunity, humoral meaning like bodily fluid, is the component of your innate immune system that is present in the bloodstream. And so the one chief element of humoral innate immunity is going to be the complement system, and we'll touch on that briefly. Next is going to be cell-mediated immunity. You guys touched on a lot of the cells that make up the different arms of the immune system. And so in cell-mediated innate immunity, some of the chief players are neutrophils, monocytes and macrophages, dendritic cells, as well as a lot of other granulocytes. But the chief players to recognize are neutrophils, monocytes and macrophages, and dendritic cells. And one of the chief activities of innate immune cells is phagocytosis, or engulfing, and then also trying to present those engulfed pathogens sometimes to your adaptive immune system. And we'll touch on that. We'll touch on that on the next lecture. So the complement system, that's the humoral innate immune system, or the chief arm of it. And so what complement is, it's a series of proteins that are uniquely able to it uses three different pathways to try and attack microbes that your innate immune system recognizes. And so the first of the three arms of the complement system is the classical pathway. And so the way the classical pathway works is there are antibodies that will recognize and bind certain pathogens in the blood, and when antibodies are binding, it's not just one lone antibody that's recognizing antigens. You're going to have many antibodies present on the surface of the microbes where they recognize that antigen. And so what happens next is there's a complement protein called C1. And so you're not going to have to memorize the entire pathway, but I will kind of highlight what needs to be the takeaway for the classical pathway. You get this multivalent protein that binds to several antibodies on the surface of these microbes, and what ends up happening is that it recruits other proteins, and it kind of clips them to create other byproducts. So you get, so you have C1 and then C2 and C4 come over. They're clipped. And in complement, what ends up happening is you get C2A, C2B when it's clipped. And when you have C4 come down, you get C4A, C4B. And so what ends up happening is C2B and C4B come together, and they create this really powerful enzyme called C3 convertase. And it'll converge to where C3 comes in, and it's just clipped. And C3 is coming in, and it's getting clipped and clipped and clipped. And C3B stays there, and C3A is kind of shuffled off to the side. But it's not unimportant. C3A is an anaphylotoxin. What that means is if you have a high enough level of an anaphylotoxin, you'll go into anaphylactic shock. A lot of cells of the innate immune system have receptors for these complement proteins. And so when these things are clipped, it's not as if they're being kind of thrown away and they're not important. They actually do serve a function. And so with the classical pathway, when you get to C3 and you're clipping a lot of C3 to create C3A and C3B, that's kind of the first critical point of the complement pathway to where you're off to the races. And so the other pathway that also is important for complement is the lectin pathway. Lectin is a class of proteins that bind to sugars or bind to carbohydrates. And so the way this works is you have mannose binding lectin, and mannose is a sugar or carbohydrate that's found on the surface of a lot of bacteria. So mannose binding lectin is synthesized in the liver, and it's found in the bloodstream. And so when you get enough mannose binding lectin to create a complex with a few other proteins that are also present in the blood, you'll get a really similar multivalent protein complex that looks kind of similar to C1q in the classical pathway. And so when mannose binding lectin and the other proteins with it bind to the surface of these microbes, you'll also be able to clip C2A or C2 and C4 to get to C3. So these are two different pathways that are able to recognize, two different pathways within complement that recognize microbes, but they converge at C3. So C3 is kind of like the focal point of complement. And it's also true of the alternative pathway, but in the interest of time, I won't get to that, but it also converges at C3. So C3 is kind of the focus point of complement, where you're off to the races if you can clip C3. What's actually kind of cool about complement is that I mentioned the anaphylotoxin part of like C3b, but what's also interesting is if you complete the entire complement pathway, what it does is it creates a pore within the surface of the microbe. It's called the membrane attack complex, or MAC. It's pretty cool. Like it creates basically it drills a hole everywhere throughout the microbe and just blows it up, which is pretty interesting. I thought so. And yeah, so two critical pieces of innate immunity that we're going to try and focus on now. They're called pathogen-associated molecular patterns and pathogen-recognition receptors, and this is kind of the key part of cell-mediated innate immunity. So first, pathogen-associated molecular patterns, or PAMPs, they are the recurrent, they're kind of the features on a pathogen that are recognized. They're called molecular patterns because they have this repetitive structure. And so whenever each receptor is kind of fine-tuned to recognize a specific molecular pattern, and that pathogen that elicits that response is a PAMP. And so PAMPs are recognized by pattern recognition receptors, or PRRs. So you have PRRs and PAMPs. PRRs are the receptors, and PAMPs are what are recognized with that specific feature of interest. Of interest. And in innate immunity, when a PAMP is recognized by a PRR, you get a series of responses. So PAMPs can also induce chemotaxis, and so I think Dr. Barker touched on that, chemotaxis being the migration of cells to almost chase down that PAMP. If I remember correctly, I think in the pre-lecture there was a pretty cool video of a neutrophil going around and it's like chasing a specific PAMP. And so that's exactly what happens in vivo, or like in us, and clinically. You'll have these cells that'll chase down the PAMP. And in response to the binding of these PAMPs, you'll have changes in gene expression, and these changes in gene expression will elicit different cytokines and chemokines. Cytokines are secreted proteins that enable communication to different immune cells, and some of those cytokines will induce chemotaxis, and those cytokines that induce chemotaxis are called chemokines. So again, cytokines are secreted proteins that enable communication in different immune cells, and those that induce chemotaxis are called chemokines. So the first classic pattern recognition receptor that we're going to touch on are formal peptide receptors. It's a type of G protein coupled receptor, and I understand you guys touched on those recently. And the way this works is it recognizes formulated peptides from bacteria. And so whenever you guys will talk about protein translation and gene transcription later on, but whenever proteins are being newly synthesized, when they're being translated, in bacteria, they're first formulated. The first peptide is formulated, and that doesn't happen in humans. And so that's one of the first examples how we've kind of our immune system has co-evolved with bacteria, where our bodies have recognized in bacteria the first peptide in a newly synthesized protein is formulated, and so our immune system has developed the capacity to recognize them. And so that formyl group is right there on that first segment right there, and so the classic ligand for our formyl peptide receptor is FMLP, or formyl methionine leucine phenylalanine. And so it's a powerful chemoattractant for neutrophils and macrophages, kind of your key actors in cell-mediated innate immunity. And as I mentioned earlier, neutrophils will kind of come to the scene. So I really like innate immunity. The best way to describe innate immune cells is that they're fast but dumb. So neutrophils will come to the scene, they'll gobble things up, and then they'll just die. And so they'll do that in the first. They'll do that in the first couple of days. But when monocytes leave the bloodstream, they'll become macrophages. And so when macrophages come in, they're kind of people who study different cells of innate immunity might argue different things. But when macrophages come to the scene, they're kind of they kind of send cues to direct what's going on. And so some people like to say the macrophage is kind of like the orchestrator of cell-mediated innate immunity. If you talk to an innate immunologist, they might agree with you or slap you in the face. So people feel very strongly who the chief orchestrator is in innate immunity. So those two cells will direct what happens in response to these FMLPs. And so again, the pattern recognition receptor is formyl peptide receptor, and the PAMP is FMLP, the peptide that's formulated on the front end. And what's interesting is that in humans, we have 3 FPRs, formyl peptide receptors. Mice have 6. Generally, humans are kind of considered like the most advanced, have the most advanced immune system. We're kind of version 10.0, and mice are version 3.0 or something like that. And so it's kind of interesting that the less advanced immune system in mice has more recognition receptors than we do. And so that's just something noteworthy. Next up are toll-like receptors. They were first discovered by fruit fly researchers. A lot of times, a lot of the first people to discover some of these genes or proteins are oftentimes in fruit fly researchers because it's a really simple model to try and study. And they named it toll, and when it was discovered in humans, they decided to just call it toll-like. So these next set of receptors are going to be called toll-like receptors, or TLRs. So TLRs are pattern recognition receptors, and they come in a lot of different flavors. And what's kind of interesting about them is that that some toll-like receptors are present at the cell membrane, and some are present intracellularly. And so I mentioned before that our immune system co-evolves with the pathogens that we're exposed to, and so these toll-like receptors are expressed. Their spatial expression in the membrane, on the membrane or internally, is dependent on what they're recognizing, and what they're recognizing can sometimes be externally or internally. And so the first few that we're going to focus on are, for example, double-stranded RNA. So our RNA is not double-stranded. Our RNA is single-stranded. But in RNA viruses, their RNA is double-stranded. Yeah, just making sure you guys can see what I see. And it's recognized by TLR3. So the PAMP is double-stranded RNA, and the pattern recognition receptor is TLR3. and TLR3 is expressed internally. And so RNA viruses will enter into a cell, hijack our machinery, and continue to create more virions and expand itself, and blow up the cell and move on to the next cell. And so the TLR3 receptor that recognizes RNA viruses is internal. It's an intracellular TLR because that's where that virus is located when we're infected. Next up is flagellin. Flagellin is it's the propeller on motile bacteria, and it's largely recognized by TLR5. And again, I believe TLR5 is an extracellular, a membrane TLR, a membrane toll-like receptor. And so it's going to recognize the flagellin on these bacteria because they exist extracellularly. And so the next three PAMPs on this list behind me, we're going to focus on in more detail. Lipopolysaccharide. So this is a PAMP that is recognized by TLR4. TLR4 is a membrane-bound toll-like receptor, and so it recognizes specifically gram-negative bacteria. Gram is a stain that's used to kind of classify different bacteria, and so you have gram-positive bacteria and gram-negative bacteria, and the positive and negative is just the ability of these bacteria to take up the stain, and so it's oftentimes the first way of classifying bacteria when someone has a really bad infection. If someone's septic, they'll try and figure out what bacteria is present in their bloodstream. And so LPS is on the surface of gram-negative bacteria. And so you have a hydrophobic lipid tail on the membrane. They're gram-negative are double membrane bacteria. And so you have a hydrophobic lipid tail. You have a core oligosaccharide and this glycan polymer that is part of the repeating segment that is recognized. And so the LPS is also referred to as an endotoxin. And so endotoxins are anything that's present in, any toxin that's present at all in the bloodstream can elicit some really strong, potent inflammatory reaction. And so a lot of times when things are purchased for biomedical research or for the clinic, they'll say, is it endotoxin free or not? And they'll put that in big letters on the front of the package to say it's endotoxin free. And LPS is among one of the most well-studied endotoxins. And so again, TLR4 is the pattern recognition receptor and LPS is the PAMP. Next up is lipotoic acid. Lipotoic acid is on gram-positive bacteria. So gram-positive bacteria that take up the gram stain oftentimes bear lipotoic acid on its surface. And so the toll-like receptor for lipotoic acid is TLR2. And so again, because you're recognizing bacteria that largely exist outside the cell, TLR2 is on the surface. And so lipotoic acid, the way it works, it has a diacylglycerol anchor in the cell membrane, in the bacterial membrane, and it has a glycerophosphate chain, and it has a polymer that contains a lot of sugars. And so the way it works is TLR2 recognizes the repetitive structure because, again, it's a pattern recognition receptor. It's constantly looking for the specific patterns that it's specific for. And TLR2 binds to lipotoic acid. Next up, CPG, cytosine-phosphate-Guanine. So I'm sure some of you might recognize a few of the terms in there. Cytosine-Guanine are nucleotides in our DNA. And what's interesting is that in bacteria, in humans, we have a lot of cytosine-guanine dinucleotides, or they exist together a lot of times in our DNA, but they're oftentimes methylated. And I think, yeah. So they're methylated by DNA methyltransferase. You don't need to know that enzyme, but what's important to know is, oh, sorry, not yet. It's been 10 years, I forgot what was important later on. Okay. So in humans, this is important. In humans, cytosine-guanine islands, these regions that are rich for cytosine-guanine, are methylated. In bacteria, they're not. So in our innate immune system is uniquely, again, equipped over generations to recognize cytosine-guanine that's not methylated. Oh, excuse me. I get excited. And I didn't mention this, the toll-like receptor for that is TLR9. What's also important to know is that TLR9 is intracellular. So our DNA is not, our DNA is oftentimes not in endosomes. Okay? Our DNA is inside our nucleus. But TLR9 is present in these endosomes where they'll engulf these bacteria or viruses, because there are RNA viruses and DNA viruses. And so in these endosomes, they'll engulf it, and within the endosome inside the cell, TLR9 is present, and it's trying to detect CpG. And so because our DNA is oftentimes restricted to the nucleus, and if it's not, that means that cell is dying. TLR9 recognizes CpG, and it's an intracellular toll-like receptor. Next up, I've not been looking at my notes. So one thing I haven't been mentioning is that what's interesting about the immune system is my perception, at least when I was sitting in your seats, was that a lot of these things were discovered decades ago, along with like Francis Crick, Rosalind Franklin, like back in the 50s. But a lot of these things have been discovered like in the last 10 to 15 years. A lot of the people who discovered these things have been awarded Nobel Prizes recently, or when you look at the papers where these things have been published, it's very recent. And among the most recent ones is C-gas. C-gas is a pattern recognition receptor that was discovered, within the last 10 years. So we're constantly trying to better understand the immune system and what different pattern recognition receptors exist to help try and find better therapies. And so C-gas is cyclic guanine adenosine synthase, and the PAMP for that is DNA in the cytoplasm. So as I mentioned earlier, our DNA is restricted to the nucleus. If it's not in the nucleus, that cell is dying. But C-gas, what it'll do is it'll recognize cytoplasmic DNA, and the way this works is when C-gas recognizes DNA, it'll undergo a conformational change, and it'll become a secondary messenger called C-GAMP. And this is a highly reactive reaction where if you get one DNA, you get many C-GAMPs in response to that. And so the next thing that happens is that when C-GAMP binds with STING, and again, as I mentioned earlier, these things are still being understood because it was only discovered recently. When C-GAMP binds to STING, you get another conformational change, and this conformational change will alter the expression of interferons. Now, interferons are among the key mediators of cell-mediated immunity. And when you get interferon expression, you are able to see a heightened response to viral infection. Almost every cell in our body, we'll talk about this in the next lecture, but every cell in our body has a protein called MHC1. It's almost like the ID card that says, for example, I'm Dan. Every cell in my body, almost every cell, should have MHC1 that says I'm Dan. And what interferons will do is they'll increase the expression of MHC1. You guys don't need to know this. This is just kind of for getting a better picture of how interferons work. It'll increase the expression of MHC1. So what'll end up happening is instead of saying I'm Dan, they'll say I'm Dan. I'm a healthy cell. I'm Dan. And cells that are infected won't have that elevated expression of MHC1. And so that's how our immune cell will recognize, okay, this cell that's not screaming I'm Dan is an infected cell, and we're just going to kill it. And so we have cells in our immune system that are targeted towards killing infected cells. But we'll talk about that next lecture. But what's important for you to know is that C-gas is a pattern recognition receptor that recognizes cytoplasmic DNA that leads to changes in interferon expression. Our last pattern recognition receptor that we're going to touch on is called NOD1 and 2. We'll be focusing on specifically NOD2 because of what we touched on earlier in this lecture. NOD2 recognizes peptidoglycans in the cytoplasm. And the way this works is NOD2 is expressed specifically in macrophages and neutrophils within the gut. And as you can see, with around it you have N-acetylglucosamine on either side, and you have this N-acetylmuramic acid with two peptides down the chain of it. And so NOD2 will specifically bind to muramyl dipeptide. So these N-acetylmuramic acid with two peptides on it is muramyl dipeptide, and that's what NOD2 recognizes. And so what ends up happening in Crohn's disease is that people have undertaken a lot of work to figure out what the genetic causes are. And so what's really interesting is that there was work done back in 2001, two separate groups published work at the same time in the same journal in the same issue. And if you notice the page numbers, they're right after one another. And so it's kind of interesting that there's a race to figure this out. But what one group first did was they did a linkage analysis. The way that works is it is an unbiased way to figure out what variations in different regions of the genome are strongly associated with Crohn's disease or whatever phenotype or whatever disease you're interested in. And so what they're able to figure out is that on chromosome 16, that there's a strong association with Crohn's disease within a specific variant. And what the next group did was they did a family pedigree analysis. And so what they did was they found a mom and a dad who don't have Crohn's disease, but have two kids that both of them have Crohn's disease. And so what they were able to do is that they were able to analyze the children's versus the parents' expression of different genes. And what they were able to see is that with NOD2, which is the same locus or same gene location that they found here, that there's a truncated form of NOD2 in patients with Crohn's disease. There was an insertion called the 320 insertion C that truncated NOD2. So NOD2 is a receptor. Oftentimes, if a receptor is truncated on either end of the protein, on one end of the protein, that might not be able to recognize its PAMP anymore. On the other end of the protein, that's probably on the inside of the cell, they will no longer be able to signal down within the cell and elicit an inflammatory response. And so what these papers reported is that NOD2, that mutations in NOD2 are strongly correlated with Crohn's disease. So what folks did next is they tried to use animal models to figure out if we alter NOD2 expression or the form of it, how does that relate to Crohn's disease? And so what they used is they generated a knock-in mice. So we have knockout mice where we just take out the entire, where we try and delete the entire gene. But with knock-in mice, they can carefully insert mutations. And what they did was they used the same mutation noted in the paper I just talked about with that family pedigree. They used that 302 insertion C allele, inserted into that mouse with a knock-in mouse, and looked at inflammation within the GI tract in response to some stimulus that creates inflammation in the GI tract. And so what they're able to see is on the left side you have your wild-type mouse, which exhibits a normal extent of tissue thickness, inflammation. We think of it as tissue thickness here in this context. And in the knock-in mice, what you see is, and this is called a jelly prep? Jelly roll prep, where they basically take out the intestines, cut it open, roll it up, so you can, it's actually kind of elegant because despite the name is not elegant. What they did is elegant. What you're able to see is you're able to see the entire GI tract in one section. So you're able to appreciate that it's not restricted to one segment like it is with ulcerative colitis, but you're able to see throughout the entire GI tract what the inflammation looks like. And in response to this chemical challenge, in the knock-in mice, you see inflammation throughout the entire extent of the mouse's GI tract. And if we go down and look further in the wild-type mouse here, there's within the crypts and the villi, that's kind of the features of your epithelium in your GI tract. There's minimal inflammation, but when you look at the knock-in mouse, the entire epithelium is gone. And so it's hard to appreciate if you don't know what you're looking for, but this paper reported that the entire epithelium is gone in these NOD2 knock-in mice where they inserted the same mutation seen in patients. And so what we're able to see is that NOD2 is a critical, it's at the nexus of Crohn's disease, where when you see this mutation, what ends up happening is that there's an overcompensation again of the adaptive immune response. We haven't talked about the adaptive immune cells yet, so I'm not going to go into those, but histopathologically, you see a bunch of these cells infiltrating into the GI tract at these lesions. So the treatment for Crohn's disease, you guys will touch on this. Oh, I'm sorry. Yes. So the normal mouse on the left is wild type. And I should have said this before. If you guys have any or not, if you have questions, when you have questions, feel free to chime in and ask. And we're almost done. So I should have said that earlier. So the left side is wild type. Wild type is usually referred to as like your normal mouse, like the expected mouse. And on the right side is your genetically engineered mouse that bears the same mutation that we saw that was reported in patients in those earlier papers. So treatments for Crohn's disease. A lot of patients are treated with corticosteroids. You guys will touch later on, I think, how corticosteroids work. But its large effect is to reduce gene expression of the key inflammatory signaling pathway. And next up, patients are also prescribed antibody, a neutralizing antibody for TNF. TNF is tumor necrosis factor. Despite the name bearing tumor in it, it's largely an inflammatory cytokine that is, there's, again, this isn't important. There's like the three horsemen of the apocalypse for inflammation: TNF, IL-6, and IL-1 beta. And TNF was implicated in the how Crohn's disease works when there's a lot of inflammation. And so some of these names you might recognize. Humira is one in particular. And so these drugs, and they bear MAB on the end. You guys may have talked about this, I'm not sure. Any drug that has MAB on the end is a monoclonal antibody. And so these MABs in particular are meant to target and neutralize TNF. So for your own edification, we have some quality folks that study Crohn's disease and the microbiome here at UVA. Dr. Stephen Cohn looks at chronic inflammatory diseases within the GI tract. And Melanie Rutkowski is a great researcher that studies immunology and cancer immunology, particularly as it relates to the microbiome. And so with that, we end this lecture. And not if, but when you guys have any questions, feel free to come on down and ask them to me. Thanks a lot.